and I would like to introduce Luis Caseñas.
Okay, hello everyone. I hope the morning has been treating you well. My name is Luis Caseñas. I'm a senior right now in undergrad at Stony Brook University, which is in Long Island. But actually, I do my research at Cold Spring Harbor Laboratory. It's around 40 minutes west. And really, I'm excited to be presenting my research that I do at CSHO and my project, the locus coeruleus, as a direct neural modulator of breast cancer. So, cool. I'll be telling a really powerful story about the connection between your brain and your body. For us, that's going to be between your brain and cancer progression. And it's important to know, first and foremost, that the nervous system very much interacts with cancer. Second, we can trace and image this neural circuitry underlying these interactions. And third, we can manipulate parts of those circuits and observe outcomes to then see and understand and function. And so we'll be working with the sympathetic nervous system today, the SNS, which is our body's system for fight or flight. It innervates organs really throughout the body and uses norepinephrine as its main neurotransmitter to modulate those organs' function. At the same time, one thing that's been established very heavily in the literature thus far is that the sympathetic nervous system drives cancer progression. Right? These sympathetic circuits that I showed you in the previous slide that innervate organs throughout the body, they also innervate cancer cells and immune cells directly. And then signaling from the sympathetic nervous system to these cells worsens our body's anti-tumor response and drives cancer in the periphery. And so, my lab is interested in breast cancer, and we use mice as model organisms to study breast cancer in vivo. The cancer cell model we use up above is E0771. It's a triple negative, non-metastatic model of breast cancer, which just means that the cancer is more aggressive and it doesn't spread to other parts of the body. So we can really look at the primary tumor specifically. And what I'm showing you right now is an image of a whole E0771 breast tumor after it was dissected, after it was cleared in a mouse. And you can see these sympathetic neurons, which are in magenta, engulfing the full tumor, which really just validates what we already know, right? We know that the sympathetic nervous system and breast cancer interact directly, and the sympathetic nervous system modulates its function. Moving on, this leads me to really the main question of this project. And what, for us, right, is if we understand that the sympathetic nervous system has this effect on breast cancer, then what's happening upstream, right? What's happening in the brain to modulate this signaling? Can we identify the brain regions that are modulating this signaling? And can we characterize their role in sympathetic mediated cancer progression? And we first need to, again, identify these regions. And we do this through a technique called viral tracing. So essentially, you can inject pseudorabies virus, or PRV, into a tumor, and you can watch it jump polysynaptically and backwards along neurons. It'll reach neurons in the spine, and given enough time, it'll also reach neurons in the brain. And you can take the spine, you can take the brain, and image them to see where the PRV ends up. And what we see after imaging is actually really quite striking. Even from afar, I hope you can appreciate the PRV signal in the spine in yellow. Blowing up the picture a little bit, you can see that the PRV is actually most heavily co-localized in thoracic ganglia 11 through 13. There we go. At the very bottom of the spine. And then again, blowing it up one more time to T12, you really see a lot of yellow PRV signal co-localized with the sympathetic ganglia. But across all of our samples, we see this same result. And we also see that the PRV